Begin.
All right, so welcome to another quarter or another year for INC. And so we're very glad to have with us today Jean-Marc, who is our newest member of INC. It's great, but not new to INC, not new to UCD and Salk, has been there for many years before. In fact, almost anyone in computation neuroscience has gone through Theory's lab. Right, and sure enough, so Jean-Marc has done great work here. One of us, I heard even yesterday about great work they have done on the modeling synchrony and the effect of say how spike timing and on correlations and activity in the brain. So it's absolutely there's great work done over the years. In addition to computational neuroscience, has done great work on rodents, and today we'll hear a lot of great stuff with what you can actually get from rodents. So Jean-Marc was at the University of Arizona for many years. We're so happy that he's joining us now here as a full member of INC, a research scientist, in addition to also being very soon the program manager at the Science Foundation. And it'll be very exciting work, so please go for it, Jean-Marc.
Thank you for here. Appreciate it. And thank you for the warm welcome, everybody. So what I would like to do today is essentially go through some results, recent results from the lab on spatial navigation in complex environments. So I'll spend some time on one particular set of studies and go into some details, and then I'll cover two more little experiments that we did. It's all published. I'll go a little more briefly on those, and then I'll finish up with what we're doing now, what I'm hoping to continue and collaborate, find collaborators for the work next. All right, so it's all about spatial navigation, and I'm sure a lot of you know that it's a fairly complex brain circuit. It's not just one area, it's actually multiple areas talking to each other. It's complicated. The centerpiece of this neural circuit is the hippocampus and the entorhinal cortex, medial, lateral. So this is where a lot of foci have been placed, but remember, it's really a complex system, right? Here is the big thing about this field is that as far as study the electrophysiology of neuron behavior in rats, people have used relatively simple mazes. Right? So a swimming pool, when the rat has to go from A to B, a T-maze, when the rat has to go straight and then right or left, and then circular maze or a Y maze or something like that, right? So this is, I'm going to say, 99% of studies in spatial navigation in rats, right? So what have these things in common? Well, first of all, they're small relative to what the rats are evolved for, right? So it's about a meter, maybe 2 meters, right? So it's not that big. Number one. And number two is, with a few exceptions, the number of decision points that the rat has to make to go from one point to another is relatively small. 1 turn, 2 turns, 5 turns, something like this, right? So really in the lab, what we're asking is, all right, this is great, and we learned a lot from these sort of mazes, these sort of tasks, there's no question about that. But is it truly navigation? Do they actually navigate? Or do they move in space, right? For sure they move in space, but do we really want to call that navigation? So a lot of the efforts that we put in the lab is to actually try to go a little deeper, right? Try to be a little bit more complicated. And the hypothesis is that spatial navigation system of the rats has evolved to process, in fact, complex spatial navigation, more complex than we've been studying in general so far. So question is, what is complex navigation? Well, it's easy to say complex, but what is it? All right, so here are a few aspects of complexity that I'm going to cover a few of those, and you'll see to what extent. So first of all, it can be complex because it's large, all right? It could be complex because it's cluttered with lots of things around, obstacles, all right? That's another way of complexifying the environment. It could be multi-scale, so you can ask a rat to go from a very small environment like a nest, right, versus something very far And so there are really two scales of navigation here. You navigate within the building versus across campus. That's different kinds of navigation we are doing, okay? All right, environments can be dynamics. Things can change and move as you're moving around. Right? People don't do that very much in the field of modern neuroscience. Everything is fixed, the rat is moving. But how about if obstacles are moving, if there are other rats around and so on, okay? Navigation has usually a cost. You should expend energy when you navigate. So when you go straight to right, straight to left, it's not a lot of energy. Right? So you can think of ways, of tasks, an environment where the rat has to spend significant amount of energy. Right? Navigation can be multi-goal, multi-decision points. Right? This is the opposite of what people do when they do foraging. Foraging, just go around, pick up some food on the floor. Right? You can think of a task, and I'll show you one in a minute, where it's actually much more complicated. Navigation can have multiple time scales, different speeds, right? Different amount of time from point A to point B. The rat has to negotiate. There is, of course, social navigation. Navigation can be with others. You can actually involve a strategy to go from one point to another, depending on other rats around you, or robot. I'll show you in a little bit. Of course, navigation requires memory, learning, whether it's explicit or implicit, prediction. You know, what is the path I'm going to take now in the future, in the next few seconds? But you have to predict based on your experience, right? Which is opposite to experiments that essentially shuttle back and forth between point A and point B on a linear track. All right, there's not too much prediction there. And then, of course, you can have cases where navigation requires expertise, where you actually use navigation strategy from another task and applying to this task. Right? And so long-term learning and skill transfer, all this contribute to complexity. All right, it has been suggested that complex navigation, not just navigation, complex navigation can actually be considered a form of intelligence, like emotional intelligence, like mathematics, like language. Complex navigation could be one of those. All right, so this is kind of, it's not an exhaustive list by any means, but this is what we would consider complex navigation. So I'm going to start with this recent body of data that we collected, and this one will hit a couple of these items. So it's going to be a large environment, and it's going to be a navigation that actually has a cost to the rat. All right, the rat will actually, all right, it's a lot of work. Okay, so what do we mean by large environment? Well, it's actually the largest in the world. We call that the mega space. It's a room. You can see a picture here with a rat here. This is a robot. I'll show you the robot in a minute. But just for comparison, you can see how it differs from other large environments. If you do a search in the literature and ask a large environment, you'll find these papers, right? And this is ours with a couple of exceptions, which you can see here. All right, so this is a 2D environment. There is 1D environment, which are essentially linear tracks, right? Which are very long also. Yeah, question.
So do you have a plan to build a mega space here, or are you just going to keep?
Yeah, that's the idea. We'll see what we're into that. Yeah, right. All right, and you can see what it looks like at the scale of a rat, right? So it's a big room. It's not like a field, but it would be nice if you got a field. It's a big room, and the rat is here. So dimensions are here for this bigger space. So just before I go and tell you about the experiments, just a few methodological things. It's just not large and that's it. We had to solve all kinds of technical issues because of the size. Okay, so first of all, we have to ensure that the rat covers this space sufficiently. In order to have place fields, you have to encourage the rat to move everywhere, being in the middle. Rats don't like that, right? We have to correct for length distortions because we track the rat in the bottom of the camera, so we have to make sure that we're fair as far as the matrix of the space. We have to overcome thigmotaxis. The thigmotaxis is this tendency for the rats to follow walls, right? The mice, and they like that. And we have to convince them to go in the middle because this is where all the action is. Sorry about the movies all right, overcoming agoraphobia, which is also an instinct in rats, not to go in the middle in a field that is empty. All right, so we have to train them to do that. we have to record and stimulate, and we have to do this wirelessly. There is no other way in that size to have a tether. It would be way too heavy. The rat would not be able to move. Right? So absolute necessity. And then we have also to have a way of, to some extent, controlling the movement of the rat in an open field. We have to convince the rat to go this direction, that place, etc. So we solve this in several ways. First of all, we train the rat to follow a robot that we actually manipulate. So it actually has two modes, autonomous. So you can see here a user is drawing a trajectory, and you see the robot will actually go by itself. We have options where we have a joystick, and we train the rat to work with the robot, which is called Spiro, to cover essentially the space, right? We design our own implants, and we do optogenetics, etc. We use a device that is commercially available from Neuralink, called Fellous, which give us 64 channels, so enough to report from several dozens of cells at the same time. All right, this is just to show you the size of this implant, which I'm sure Gert is going to laugh at. But this is what we have to deal with these rats, right? They have to carry this like a hat, right, around. It's Wi-Fi, and that's what we're doing. All right, and we can do it. It won't work. Cost. All right. So just a little bit about Spiro and the rats. We of course have sessions where we don't have the Spiro, the little robot, sessions where we do, so this is foraging with or without Spiro. And bottom line is that the cell properties are essentially the same, so we merge all the data that we obtain from sessions with or without Spiro. You can see here that we can get very long sessions, a kilometer worth of travel with Spiro, which typically we don't do with the rats spontaneously, they get bored and they stop, that's the point. And then we can control the speed of the rat, which is actually pretty cool. There's no other way of asking the rat to go at a particular speed. You can't train the rat to go at a speed. You have to have these little devices allow us to do that. You see that we can be pretty precise. All right, so anyway, what I'm going to show you is essentially a merge of these sessions where the rats are navigating by themselves in megaspace, and other sessions will use the robot. All right. So let's start with some very basic behavioral results here, just to make sure that you realize something about rats and space. Right? So rats are not like us. They're small, and their vision is not that great, okay? So here's a very, very simple experiment where you release the rat somewhere in the room. So this is the mega space and 39 little points that we discretize, right? So, and we release the rat somewhere, and then there is a target somewhere else, which is a little cup with a food on it. The rat is hungry, so it wants to go find the food. And the first thing you see across multiple trials is that the rat doesn't go straight. There's no such thing as, the rat should be able to see. We see it, right? It's not hidden or anything. You can see that experiment here at the bottom. And so you notice that there is many ways for the rat to go to the food, and it's rarely straight. All right. So what you notice is that there is a point at which the rat will just go somewhere, and then all of a sudden, as if it realized where the food is, will start to converge onto it. All right. And most of the time, it's about 60 degrees. So you'll see why in just a second. Question?
Yeah, there is some control of the air inside, because you know that the rats follow the smell of the food.
Yes.
And then they follow what is the direction of the smell.
Right.
And you see, and most of them is going inside close to the wall. And then that means that something that for me that is reflecting all the smell of there, and then
that's a good question. We do control for this. We have evidence, and I'm not showing you here, that the smell is not a big factor unless the rat is very close to the food. We're using food pellets, which are compacted, and we have clear cases where the rat is actually going by the food without actually getting it. So I'll come back to that a little later if you like, but smell is not a big issue here, and it's
because it's saying that most of them is close to the wall, not that.
Yeah, this is deep access. This is what rats like to do. They like to go through the wall.
So in total, they were always coming to trials.
This is just an example, right? We release the rat at multiple start points. It all differs. So the start here is the start, and this is the goal. And you can see that, there is no such thing, such a thing as a
So the
straight line.
total number of trials were in front weeks mostly.
This is just an example. It doesn't matter what the number of trials.
Um,
All right, so now you go in and you take, multiple rats, multiple paths, large numbers, and you plot this relative to the target. So 0, 0 is the target. And we can see right away that it's a big mess, except around here, all right? At which point it becomes more or less straight, all right? So what's happening, we think, and that distance here is, by the way, about 1.2 meters, all right? So bottom line of these experiments, behavioral experiments, is that rats really care or see or detect, or you call it whatever you want, but they actually go to the reward directly when that reward is about 1.2 meters away, whether it's their eyesight, it's a lighting condition, their strain, whatever it is, well, it's the areas. So this is our basic theory here, is that in this megaspace, there is a particular area in the middle, 1.2 meter away from the boundaries, where navigation is probably very different than in this area. Why? Because in this area, the rats can see and detect very precisely the walls. All right, here they can. Or this is our hypothesis. I'll show you some evidence that's true. All right, and then the side note, of course, is why the 60 degrees that I showed you before. Remember, rats are not like us. They have lateral eyes. All right, so the head direction is not really the direction with which they absorb visual information like us, right? They actually did at best visual inputs 60 degrees to the side. All right, keep that in mind. Yeah.
So you have three rats. Are they distinguishable by this distance?
No, they're just about the same. Yeah, there's some strains and age, right? And this is just an example.
Yeah.
Three rats, but all
I mean, I could imagine yours would be different than mine, but
right.
they're interesting.
All right, interesting thing also is that there is a bias, right, which we know from, rats have handedness. You have right-handed rats and left-handed rats, right? And most of them are right-handed, and you can see a little reflection of that in this, the last turn they do before conversion.
There's some process there as well.
Yeah.
The learning process.
Say again.
The rat, do they have a learning process or is new, the environment is new?
Oh, no, they know the environment, they're familiar with the environment, but that experiment, there is no learning. Every time they're released, the position is new, so they cannot learn. In this experiment, I'll show you another one where they learn. This is just purely detection, not learning. All right, so this is behavior. We talk a little bit about electrophysiology. So what we do, as I showed you before, is we record from these neurons, right, in the hippocampus. And the classical, finding I'm sure you've heard of is this finding of place cells. This is a beautiful piece of work done by many people. John O'Keefe, of course, received the Nobel Prize in 2014, and this is part of his lecture. It shows you here lots of place cells that are selective to particular parts of the environment. And this one is selective to the upper left corner, this one to the lower right corner, and every single place cell is selective to a different place. With a few exceptions. You can see here, this one has two blobs. This one has two blobs, right? Which is usually thought of as noise. It's biology. Sometimes you have multiple, field, but by and large, every place cell has one place, right? That it's selective to. Well, if there is only one slide I want you to remember from this talk, it's this one, all right? This is the finding. So this is single cells, 5 of them recorded in a small environment, a normal size, 1 meter or something, mega space, and then back to the same environment. All right, and what you can see is that that cell has a place field, like the previous slide. But as soon as you go to mega space, the place field splits. Now it has 4 subfields for this one, 5 for this one, 4, 1 big for this one. That's the exception. Right? Only 1 field.
Or it's two very close to each other.
Sorry?
Or it's two very close to each other.
They are very close to each other.
No, I mean that one big one. kind of looks like it's due.
Oh, yeah, right, it's possible, right? So we have, of course, some errors in detecting and so on, yeah, it's possible, right? All right, and then if you look at the distribution in megaspace, most of the cells have 3 or 4 subfields, all right? And in a small places, in a small environment, most of the cells have 1 field and then maybe 2, but not much more than that, right? So bottom line here is that in megaspace, the very same cell that has a single field in small environment splits and have multiple very reliable fields in large space. All right, it's the same cell, 1 cell, not 5 cells, just 1 cell. Okay, and what else do we know? A few other results. If you just plot as a circle across all the cells we recorded, the size of the fields, right, and where they occur, you notice that it's more or less random. All right, and this is a foraging task, by the way. So this is a foraging random movement of the rat, and the fields are random. And with a small tendency for the fields, for the small fields to occur near the boundaries. All right, and how far from the boundaries? Well, it's about 1.2 meters. So this is an actual physiological evidence that there is something special about the middle field, right, versus the size. Okay, so it's really important that the megaspace is actually large enough to include a sizable area, which is 1.2 meters from the boundaries. Right?
It looks like a tennis court.
Yeah, right. I mean, for a rat, it's about the same, the size of a basketball court for a human in this megaspace. Yeah.
So, like, I see the point of like having smaller subfields because probably they are more,
Right.
more sure or like more detailed, and also they
Precision.
have an anchor, which is your boundary and like some markers there. While when you're in the open, you really, like even in this space, which is a mega space, it
Right.
still is very empty.
Right.
So now it's more ambiguous about where you are.
Oh, keep that idea in mind. I'll come back to that. It's like you use the right word. You'll see it in the. Yeah, absolutely. So it could be a resolution issue. Yeah.
So Ketchum has
Yes.
an old paper, you know,
Yeah.
where he showed that, you know, what's the applicable size? It depends on the dimensionality you're trying to cover.
Right.
And for high dimensional spaces, bigger is better. For 1D, you know, small is good.
It's going to.
2D is actually the transition where all sizes are good.
Yeah.
So you want the distribution of sizes.
You do exactly for efficiency purposes.
Yeah, for efficiency.
Yeah, I'll come back to that in just a second. We're collaborating on the grant together. I'll show you something else. All right, one thing I should mention that these subfields are non-directional, unlike in the track, right? So it doesn't matter where you come from, which direction, the subfield will be activated. All right, some interesting properties about the subfield is that there seems to be some sort of relationship between the number of subfields and their average size. All right, so if you only have 1 subfield for a cell, it's large. And the more subfields you have, the smaller their size. All right, so that on average, if you do the sum area of all the subfields, it's actually pretty constant. There's variability, right, across multiple cells, but they seem to center around a particular value. All right, interesting, right? So there's some sort of homeostatic adaptation of the number of fields and their relative size, average size. All right. And then what you can do, and what we did, is vary the size of the environment, right? So big space is here, but then we can move the walls and make it a little smaller, medium, or small, and then you can see that, well, by and large, the rule occurs, the rule holds, is that for each of these different sizes, the average sum area of these subfields is more or less constant and relative to the size of the environment. Okay, so that's pretty cool too. So it's kind of an example of context-dependent coding of space. It depends on the overall size of the environment. Okay, and this is the same cell, right, across these 3 conditions. All right, so this is for multiple fields. Question is, well, can you manipulate a single field? And people have done that before, so this is not new, but we tried to make sure that we're compatible with other works. So this is what I showed you before, the small environment, and we went with very small. For that, we need a special robot, a mini sphere, which is essentially this relative to the size of the previous robot that you just saw. The rule still applies, all right? So the fields are going to be actually change shape, they're going to be smaller, which is something that O'Keefe and Burgess have noted before. So this is when you stretch the environment, you stretch the field, right? So basically, as the size of the environment grows, place fields tend to enlarge, and then at some point, they split. And then you keep on going and split, all right? So this is what we call the soap bubble theory. So this is a small environment with a place field, all right? And then, so this is a theory, a hypothesis, right? Which is compatible with what I showed you before.
All the measurement you did was when the rat followed the road.
It's both, yeah, right.
Yeah, because in this case is you reduce the dimension because the rat only uses the robot and it don't care about the.
They actually do. I'll show you some evidence of that later. But we mix both. We do both of experiment with and without the robot. We use the robot at the beginning to make the rat used to the environment, and then at some point we just let the rat outside.
Okay.
And we measure the data, so it doesn't make a difference for the robot. It's practical. All right, so this is small regular space, and then what we do is we go to a little larger space, and the fields are starting to now being distorted as you would if you have a soap bubble and you start, you know, with a little circle thing, you just move across, and then at some point it'll split and split again, right? And then this captures essentially that idea that the overall volume, the size here, is getting smaller and smaller. Of course, it's the air, right? You don't add more air when you split them on. All right, so this is a little bit of our theory. It explains the multi-scale, multi-field phenomenon with the fact that the average sum area is about the same. And it introduces another notion that we're just throwing out there, the fact that it's really likely that these subfields are anchored either to each other or to the walls. And that determines their position, right? So this is just a hypothesis, we haven't proven that. All right, and since this is Institute of Neural Computation, I just want to do a little computational digression here. So what I just showed you is this, is that if you're a decoder now, if you're watching that cell, if you're reading the cell output, and you see that cell is being active, you actually don't know where it is, where the rat is, unlike a traditional place cell where we have one place field. So one single CA1 neurons fire if the rat is at one of 4 possible locations in this particular case. So it's a hypothesis, right? So that points to something that we like to call an ambiguous code. All right, and you're going to say, well, ambiguous is not good. You know, it's a bad word. We want to disambiguate things. We want things to be clean. We don't think so. Actually, that is a very good thing in the brain to be ambiguous. But we need to come back to that later. And it's not new. So here is an example of a classical experiment, and this is an obscure course, where you send a frozen noise to a neuron in vitro, and then you can see these beautiful alignments, very precise, reliable, right? With exceptions. You see the little spikes coming, you know, here and there, right? Which, you know, in the science paper, that was noise and it was really not a big issue. So together with my colleagues here, Peter Thomas, mathematician, Paul Tessinga, physicist, myself, in Peter's lab, Peter said, you know, this is 25 trials, let's go to infinity. He's a mathematician, so infinity, 3, 4, 5 equal, these are the symbols. So he said, and this is the infinite number of trials. Right? All right, and Paul, a physicist, said, okay, you know these squigglies that we send to the neuron, you know, let's look at the frequency content. This is spectral analysis. This is physicists, right? You like to do this.
By the way, the famous book title is 1, 2, 3 Infinity. It's written by physicists.
Yeah, it's a crossover, right? All right, and the computer scientist, which is me here, said, you know, this is too much work. Let me write a computer program that makes a big loop. So I can go and have tea at 3:30 every day, and then, you know, let the experiment run by itself autonomously and so on. So instead of 25 trials, we went ahead and did 150, all right, in the same condition. And what you start to see now is that these extra spikes, all right, which occurs, you know, from time to time. So you see these beautiful alignments and these additional little things there. In fact, you can cluster the trials. So reorder these rastograms and find two patterns, discrete patterns. Right? So now when the cell is spiking in two different ways for the same input, the input hasn't changed, but it spikes in two different ways. All right? All right, so this is pretty cool. This is only 5 KFC neurons in vitro. All right, then we went and looked for data from Unwritable and Query. Unwritable was here, and they had data from the LGN and they published that. So this is what they saw. This is what they published. And we asked them data and applied the same algorithm, the fuzzy clustering algorithm, to reorder the trial. And we found that in fact, across all these 80 plus trials, there is like between 2 and 3 groups of trials that are reliable and precise, right? Not just 1, but 3, okay? And then we went around and asked, begged for data from Albright's lab and found monkey NT data of a monkey. So this was a cat, an anesthetized cat, recorded in LGN, watching full field movies, right? It's several seconds long. This is a monkey awake monkey looking at a screen, looking at little gamma patches moving. And same thing, you see these alignments, but within these alignments, you see up to between 2 and 5 subgroups. Right? So this is like very similar to this, we think. Right? So the same cell responds 5 different ways.
It's worth saying that the stimuli were very special in Brandeis cases of flickering.
Right.
I think dot, but in the case of, it was a Gabor in MT, which we like, but it was, randomly moving up and down across its, the wavelet, the Gabor.
But so the current, it's a phase shift of the.
The phase shift, it's like a phase shift, yeah, but but if you don't do that, then you just get firing rate.
Right. Okay, so this is kind of different ways of illustrating that idea of being ambiguous. So the same cell responding different ways to the same stimuli, whether it's in vitro, in LGN, or in MT, or in the hippocampus. But, here, what is constant here is the environment, not the waveform.
And can I ask a clarifying question?
Yeah.
What's the difference between C and D on the left? It's just, I know.
This
that-
is the same trials, the same histogram, but reordered. The trials are reordered to put together, to illustrate, to show the 2 clusters. Right? But it's the same trials. It's just like, they occur randomly in interspace. All right, so let me summarize this part, and I'll go a little faster in 2 other experiments. So conclusion of this body of work. So dorsal C1 place cells are multi-field, multi-scale in data space. This is in fact the rule, not the exception. We rarely find a dorsal C1 place cell that has only 1 place field, right? Opposite of the complete and clouding results. In this environment, they have an average between 3 and 4 subfields that vary in size by an order of magnitude. So you can have a small subfield and 10 times bigger subfield in the same cell in data space. Coverage of space is generally uniform and random with a question mark because the task here is random. So it would be something about the task. Point. The larger number of subfields, the smaller the average size. And the sub area of the subfield is constant on average and commensurate with the size of the environment. Right? So, but bottom line, single cells are multi-scale and code space ambiguously. The code is spatially holistic. Right? And all this has been published. If you like to see it.
So you call it a megafield, but in comparison to the natural range that the rat is in the wild, it's tiny.
Yes, that's true. It's as big as we can make it.
Yeah, well, you've gone as far as you can in the building that has walls that are, 12, 20 feet apart. But would you speculate if you could record from a rat in its natural environment?
It would be amazingly interesting to do, and I'm hoping we're going to get there eventually, because we have the technology, the wireless system, right? And my guess is that you're going to have even more subfields.
And they get smaller and smaller.
And it's gonna get smaller and smaller.
And so what happens when it goes to infinity?
No, it's a great question.
Yeah, so the bubble, you said, are you increase the space or you have some anchor on the borders and that's why they separate, that's your theory.
Yeah, that's the theory.
That's for a rectangular environment. What happens if it's Yeah.
I don't think it matters, but it's a great question. We haven't done it in any other way. I mean, it's a great thing. There are boundaries, no matter what. Right? So whether the boundaries are circular or it shouldn't matter as far as the field is. But I don't know for sure.
How does it know where it is if it can't, it's in a.
Yeah, good point. So there are cues in the room. So
Right, you said
well, they can see blood. They probably don't see.
they know how
details.
far away the end is,
Probably.
basically.
Yeah.
Flags of
They don't bump into walls, right? They're not that bad.
So this is getting mapped, like, when they start, that's when they map.
They know the room. The room doesn't change. They're familiar with the room, right?
And it doesn't matter where they start.
No, right. The room has big cues, national flags from manipulation.
Just listen to the anchors.
Yeah, so this is what makes them feel, I think, I'm just, hypothesizing here, that make them feel they're in the same environment. Nothing has changed. It's the big room. It's the main aspect. The robot, the food pellet is that size, right? It's a couple centimeters. It's much smaller. That's the part where they need 1.2 meters to see the goal, right?
Do you see any like discernible features that are shared between the different locations of one place now, or is it kind of random?
No, it's, as far as we can tell, for this task, it's random. There is nothing special. Now, we have other tasks when we see aggregation of the place field in the reward site, we see border cell, we see the object vector cell, all of that, as other people do. Yeah. Or here, yeah.
So place cells are different in the magnet space. Are grid cells any different?
Oh, great question. So this is Grant who just got, and so that is recorded there.
Great.
Yeah,
But
so we don't know.
Grid cells.
Grid cells, right? Grid cells are multi-field also. It's another example of an ambiguous code, because when the grid cell fires, you don't know where you are, right?
Right. So, are there any motor cues?
No, so that was a question that we had before. So we control for that. Smell is not a factor
Okay.
in this space. It's, first of all, it's large and there's a lot of air circulation.
So that's
All
like,
right,
because it's,
sorry.
they're mapping it based on visual, it's not like how many steps it took to get.
Oh, that would be amazing. if They can count the step. No, it's, yeah.
So
I doubt.
visual and yet not good with vision.
They're not good with vision, unless it's pretty close to 1.2 meters away, in which case they're okay. All right, so let me go with it quickly now. In a different experiment is published, I don't want to explain too much details, but a different kind of experiment where we try and hit a different kind of complexity, cluttered environments. Navigation has a cost. Navigation has multiple goals in this experiment, and it requires memory. So this is a memory task. And this one is focusing on a different aspect of the hippocampus, which is its dorsal ventral axis. I don't know if you know that, but the hippocampus is a C-shaped structure, which is like this. If you look at the left, the nose of the right this way. And most people have put the theory forwards that dorsal and ventral, in fact, are doing two different things. Spatial navigation in the dorsal and other things in the ventral, right? And we think that's actually not entirely accurate. Okay, so cluttered environment, it's like a campus. You have to go from point A to point B. There is a lot of obstacles, buildings, streets, etc. Right? The basic issue that we have with these studies of dorsal ventral hippocampus is that they're all done in the kind of environment that I showed you before, small, right? Teammates and all of this, right? And then we also have a series of papers that we published with collaborators showing that having multi-scale coding is actually good from a modeling standpoint and memory recall and storing, etc. I'm not going into this, but we started essentially by modeling that here and then started to ask the question, experimentally, does it make sense? So the hypothesis is that the joint involvement of dorsal and ventral portion of the hippocampus is in fact required in complex, in this case cluttered, that's the complexity environments, right? So what is the task? So it's a task in a small environment here. So the size is not an issue. It's a relatively 1.5 meters, where there are 8 feeders at the periphery. And the rat has to discover that only 3 of those are baited. We're gonna get sugar water, right? So first phase of the experiment is totally confused. It's going around, where is my sugar water? And eventually it's going to find that only a subset, this one, and this one, so 3, 5, and 7, if you count 1, 2, 3, 4, 5, 6, 7, 8. And then it's going to learn that, all right? And then it's gonna go now without, goofing around from one to the other and get reports, randomly, right? In all of these.
In this,
All right,
in this case, they follow the eyes, right?
they follow
The sea.
they can see, yeah, this
And
is
this
small,
is,
it's 1.5 meters total diameter, so wherever they are, they can see everything. All right, then they go to sleep. Let me just finish this as the movie is going. I'll take a question. And then we ask them to recall this now with obstacles, Lego objects, right? Then you can see that at the beginning, even though it's exactly the same features, the rat is a little bit confused now. It has to recompute its trajectory all around. Where am I going to go? Which is the path, right? But eventually, they will actually find. They will redo this 3, 5, 7 pattern and converge and find, wiggle their way around the obstacles, right? So that's in recall mode. So they learn first without obstacles, and then recall with obstacles, right? And then we can also do the same thing now with large obstacles, which you're gonna see in just a second. Because we thought, no, dorsal, ventral, small place field, large place field, maybe the size of the obstacle matters, right? So it's turns out doesn't matter, so we'll skip. So this is the experiment. They learn a set of features, right? It's different every day. And then we go ahead and do an inactivation study. We essentially turn off the dorsal or the ventral using an anesthetic bilaterally, right? Which lasts for half an hour, right? So we turn that. And we ask them to recall. So either we inject the anesthetic or we inject saline as a control. And then we have small obstacles and large obstacles, and all of this is mixed up, right? This is dorsal hippocampus, this is ventral hippocampus, which is really intermediate in this case. All right, so I'll show you the basic results. If you don't have any obstacles, whether you anesthetize the ventral or anesthetize the dorsal, it really doesn't matter. Those are 3 different measures of behavior. So the number of times that they go to cups that are not baited, the total distance they travel around, and the number of times they get confused and stop. So 3 different ways of measuring behavior. So if there is no obstacle, doesn't matter, dorsal or ventral, same performance. And here's the surprising part. Now, if you have obstacles, small obstacles, then you can see that the behavior of the rat is the same, whether you inactivate ventral or you inactivate dorsal. Okay, the theory was that ventral does not matter. You can inactivate ventral, the rat should behave exactly the same way. Not if it's complex. So yes, for the previous slide, no for this one. And same for large environment. Okay? I'm sorry, same for large obstacles. All right, so summary conclusion of that one. I'm sorry, I'm speeding up a little bit because I want to show you a few more points. Inactivation of dorsal and ventral does not impair trivial no-obstacle spatial navigation, and dorsal and ventral are both involved in obstacle-rich environment, unlike I said before. And in this particular experiment, the size of this obstacle did not matter. All right, so our hypothesis is that in complex environment, with the kind of complexity that I showed you here, the coordination between dorsal and ventral is crucial for being efficient. For finding what we're calling finding results.
It's possible that the neuron decide to go to the hippocampus and go directly on the processing is in the cortex. Is that the reason they don't detect something or else?
Well, I mean, we see an effect when we inactivate the dorsal or ventral. So we know that dorsal hippocampus and ventral hippocampus are involved. Right? It's not just cortical. Cortex is involved for sure.
Yeah, sure.
That. Oh yeah, for sure. All right. So last one is large environment navigation that has a cost, navigation with multiple goals. All right. So imagine that you have to go through 5 different places, movies, a farm, etc. You have many ways of doing it. And you have the non-efficient way, which is taking a very random route, or you have to be very efficient, find an optimal route through these 5 little places. All right, so this is a classical problem in computer science, so the traveling salesperson problem. There's a lot of things we published on this from optimization from this point of view and computer science books it's called an NP hard problem, because it's not trivial. If I give you the position of the cities, there is an equation that you can give you that defines the optimal path. You have to either go through all the possible combination and sort them and find the smallest, or use heuristics, which doesn't guarantee that you find the smallest. Humans can actually do this. There's been some experiments showing that you can find an optimal route. Animals can do that too, so it's been tried on chimps, on marmosets. I'll come back to marmosets later. And vervet monkeys and even pigeons, when they peck on the screen, they can peck efficiently through a few points. Right? So I'm going fast, and then of course rats. So this is what we try to do, is let the rat off a particular place in this environment with 5 rewards. And at the beginning, the rat really is confused and go pick up all the 5 rewards in no particular order, but at some point, they will converge into a particular path that will kill. And we stop them when we do that 3 times in a row. So we actually find an optimal route, which is usually within 10% of the absolute optimal. So they're pretty good, and you see a little example of rats in a large environment trying to find these rewards. And remember, they have to be 1.2 meter close, right? So you have to remember from the previous trials, 6 trials, if there's more, what is the best path?
So from, I'm not too sure if this is just an example, but like in the real experiment, have you done it, or what would you think will happen if the position of the rat is shifted by like?
Yeah, we did that.
And
We did that. They get there. Yeah,
Oh.
They eventually find the optimal route, which includes the start point.
Oh, okay.
All right, so electrophysiology, so we did that with electrons in the hippocampus, and we're trying to find how do we know that they're optimizing? There's something in the place cells in the hippocampus that tell us that the rat is working to optimize, right? The key here, we think, is this phenomenon called the shockwave ripples, right? So from time to time, if you look at the LFP in the hippocampus, you see a beautiful theta oscillation. So just a second, the rat is going around, and then all of a sudden you see these awake shockwaves, which are very fast oscillations that occur from time to time, entering the cells in the hippocampus, which occur at multiple points when the rat is moving around and finding its way. So this is the rat learning, right? And what we find, so this is a shockwave, and you see some neurons firing at the same time, right? This is the visual results, is we find that the number of shockwaves per trial is the highest when the rat is trying to optimize, right here, right? It is lower when the rat has found the optimal path already and just converging onto it, repeating it, right? So by memory alone, for instance this. And it's the lowest here in the condition where we actually lead the rat through the different rewards, artificially with a robot, using the path of a different route. So there is no learning, no optimization, we just follow the robot. Right, and of course, if the rat is just foraging, then there is very small number of shockwaves also. So in our hands, it looks like the number of shockwaves on the shockwaves is in fact the key to study optimization. It is probably where all the action is within the shockwave. What do the cells do? So we looked at this from the modeling point of view. We try to model this with colleagues, me and who was in Fellous. And we tried they tried to implement as much as of the different gray areas as possible, including the PFC implemented as a reservoir, the hippocampus and the reward area, the striatum. To cut a long story short, we essentially, the way this works is the hippocampus generates a little hypothesis for each of the shock waves, generates a sequence of places, very short, not the entire path, but a small one, sends that to PFC, PFC reverberate and send that back to the hippocampus, and little by little, as the place fields overlap means the distance is short, right? And this is where the shortest path is found. The shorter, the more the overlap, the shorter is the path, the more reinforcement it gets, and then you eventually find the shortest route. Right? By trying non-optimal segment that we call snippets, you actually find the optimal path. And by the way, Jean-Marc was the first one to actually do this in the neural networks a long time ago. All right, and I just want to spend a few minutes just telling you what we're doing now, the kind of other complexity or kind of other experiments we're hoping to continue. All right, and of course collaboration to establish. This is the first one. So this is the TSP now in a dynamic environment. So multiple autonomous robots just going by themselves, and then the rat is trying to find the optimal route, which is across these little black cups. You can see that it is interfered with these little robots, right? But the rat is doing a good job. It's actually going to find, to recompute dynamically these trajectories as it is interfered with by these robots. All right? So this is a case where the complexity.
How are the balls controlled?
This is 8. We went up to 16.
No, but how, the direction, yes,
what's the control?
directions or just random?
Oh, this is random. This one is random.
I have one of these balls for my cats.
Yeah, exactly. It's a cat.
It's for, okay, so you made this call that, you know.
No, we didn't make that. We actually tuned them so they have the same speed of a rat, and then they do turning as many times as normally a rat does when foraging. Right? So we actually modified it a little bit so that it mimics movement of a foraging rat. Right? All right, so spatial navigation optimization in a dynamic and unpredictable environment. And that calls upon other brain areas and other types of cells that are interesting. So the lateral internal cortex, object vector cells, landmark cells, boundary cells, all of these are important in this experiment. We just got a grant with a Jean-Marc and
Why is it going to the black balls? Are they food? Is that food?
these guys are just cups. They where the food is.
They're
Yeah.
food.
They have to collect the food from these cups. That's their goal. That's their task.
Apparently, that the rat is not important, the balls.
Yes.
When they follow the goal is to get the food.
Yeah.
And don't care about the.
Well, they do care because it comes
Yeah,
to
but
that.
you look,
This
when
example,
people
right?
do
But you can see the path of the rat, right? It's not straight from cup to cup. It's actually, a little bit like this. And they have to actually optimize. They have to make an effort.
No, but when you come back, yes.
Yes,
for
that
the-
too. Yeah.
Follow the balls, but when looking for the food, I don't care the balls.
Yeah. Well,
At least in this.
they will not. They will not follow the robots, right? They will ignore the robots, but they have to do it in a way that doesn't perturb their movement. So that's the key. They have to negotiate, like you're doing when you're driving, right? You drive from point A to point B. There are multiple cars going in front of you. You have to, wiggle your way around. Same thing. All right. So as I said, we just got a grant to look into this and other studies with these collaborators. These two are in India, and this is the DST, and NSF is funding the American part, which is me and Jim here. And then we just won a grant with Andrea Shiba and Kenchen Zhang that they just mentioned for NSF under review right now to look at something below the dorsal ventral axis of the hippocampus and Lisman. All right, and if you thought that TSP in negative space is not large enough, it's not hard enough, try 3D. All right, so this is now TSP, traverse person problem, in 3 dimensions in an environment which is essentially a garage structure. All right, classical problem, people losing their car in a garage, you know where they are. And we think this is about reasoning in 3 dimensions, right? So rats are not 3-dimensional like bats, they're 2-dimensional, 2.5, right? Like us, we can work with multiple levels. But the big starting point for this, for us, was like a very simple experiment. Going to a building where your office is on the 3rd floor and ask your guest to point to the entrance of the building, right then. Like, point, entrance, building, down. Well, okay, if you do that, because we're on the ground floor, right, that's easy. But if you were on multiple floors, you'll see people actually are confused, or at least I am, right? So the question is, can you reason in 3D? Can you just imagine the reward sites, which are in black, in 3 dimensions, and then find the optimal route?
So I thought that rats burrow into the
Yes,
ground.
they do, but they
So
also
they do
climb.
have multi-levels.
They do, yeah, they can do that, and they also climb. They climb on things, right? As you can see in this little movie here, they do that very well. So in this experiment, this is a garage structure, but there is no way of going from this reward to that reward. What you have to do is go all the way around, and so it's very inefficient. You can see the rat do that here.
And did they take an elevator to go up?
No, there is no elevator, but they can climb, right? And this is what happens in this ride. They actually find after training that the optimal route through all these 8 rewards is what?
Going up.
It is going up, right? It's actually climbing. 1, 2, 3, 4, 5, 6, 7, 8. Right? They don't do that in
garage structure here, right?
yeah, but then you have to take the stairs, yeah. But the reason why we're excited about this is that first of all, it's about thinking in 3 dimensions, right? Navigating in 3 dimensions in different ways than bats and different ways than hummingbirds and whatever. It's now multiple 2D maps. Is there remapping? How are the place fields essentially in one level or another, right? And in this particular experiment, we're stuck. We can't do the recordings. Why? Because first of all, we cannot do that tethered. There's absolutely no way you can have a cable linking the rat, the recording device to the computer. So it has to be wireless. And our wireless device is that big. There is no way the rat can go in between these floors. And if you make the floors bigger, they refuse to climb. So we cannot do the experiment unless it's lightweight, small, and long lasting, right? So this is, I'm hoping, collaboration with Gert's lab. And we will want to try and find a device that can help us record from this things. All right, and I want to finish with what I think is the next step in these type of experiments. We're reaching a point where we're really thinking too complex environment. It's too hard for the rats. We want rats to do way more complicated things. And so we need to shift to something else. And then we think the marmoset. And of course, beautiful work from the CSD here, Corey's lab, with Gert, and others reporting of that. Found that the marmoset is a fantastic preparation. They have place cells, there is sleep is important, so nice sleep patterns, spindles, shock waves, etc. Interesting difference with the rodent in terms of the theta oscillation, it's intermittent, this is the lab. Spindles, etc. And we're collaborating right now. I've been spending a year essentially working with marmoset with a colleague at ASU, Arizona State University. It's so much fun to work with them. We have no idea. A recording from the auditory cortex in this case, and we have some interesting results. All right, lots of people to thank. The ones circled in red here are the ones that were involved in the experiment that I showed you, but there's really many people we're collaborating with, postdocs, graduate students, and collaborators. And I just want to finish with a few other movies, a few other things that we did, we're doing. Well, that we did start. So hopefully the movies will show here. But this is just to give you a flavor of what you can do when you have an AS space, right? You can now address many other scientific questions. For example, one-shot trials where the robot is showing the rat where the food is in a very complex environment. And it takes only one trial for the rat to actually go by itself now and find the reward. So this is proof that they pay attention to the environment even though they follow the robot in one trial. Robot-assisted path learning, they actually follow the exact path. There is mimicry, where there is an equivalent way to get to the reward, but they tend to mimic what the robot did before. This is our latest attempt at autonomous robot that actually sense where the rat is and adapt its speed and direction with respect to the rat. So there's fully autonomous. When the rat is too close, they speed up. When the rat is too far, they slow down, etc., etc. So we have a lot of robotics.
What's in it for the rat?
Oh, there's a bit of food at the back. Oh, yes. Yeah, so the robots are positively motivated, right? So they're not dangerous robots. There's no moving parts, right? We hide everything. They don't make any noise. We work very hard in having robots that are compatible with the rats. Right? And we actually have doing a lot of work on this too. All right, and then I'll just stop here and take any questions. Thank you.
When you did the obstacle,
Yes?
When you did the obstacles, did you see any, do you end up analyzing the sharp ripples and realize that there were more sharp ripples because there's more demanding tasks?
Yeah, no, we haven't. So the obstacle experiment was an inactivation experiment, so no recordings. But this is definitely the next step. We should do that. Yeah. Right.
Something I was wondering about during the same experiment, basically, so the animals learn in the environment, and then you add obstacles, which should in some way lead to a remapping of play cells or
Right.
whatever, so how could they possibly apply what they've learned before if it's reference to play cells?
Remember, it's the same rule. We have cues out there, right? Like in the Morris database, right? There are cues around the row, so it's not totally unfamiliar to that. The actual detail of the navigation from going from this point to that point has to be recomputed because of the obstacles. They eventually do that, right? It takes a little bit of time, right? But they readapt. Question is, do they react? I mean, are there like new object vector cells now bound to the obstacles? Are there, are shockwaves, more numerous when you have to make that effort to recompute, which makes sense, given the other things that are shown. So all that is another question we just need to do.
So, as you go from dorsal to ventral, the facials get bigger. But I think that if you keep going ventrally, you eventually reach a point where it's qualitatively different. In the sense that it has connections with different connectivity with other parts of the prefrontal cortex. And it's thought at least that it is serving a different function, right? But do you ever explore that or?
No, it's true that anatomically you have connections with PFC, the amygdala, with the ganglia, all this more ventral than dorsal, right? And also neuromodulatory centers. The other thing we're saying is that in addition to this, it's also involved in spatial navigation. It's not exclusive. They're multifunctional. The cells in the ventral hippocampus are involved in multiple things, including spatial navigation in complex environment. So what we think that what the results are showing is that the ventral is about complexity, it's about other things, amygdala, so emotional things, planning, complex environment with obstacles, emotional here, size, whatever. It's dealing with complexity more than it is dealing with simple memories or.
So, it's interesting, maybe one way to think about it is that when a rat really has to go a long distance, has to plan, and that requires other sources of information. Is it the dangers there? I have to go through a danger zone, right? Maybe I should avoid some places. And maybe that has to be added in, as you say, to the representation so that you can.
And that's ventral.
Yeah, the ventral adds that, yeah.
And by the way, ventral dorsal are in interactions quite a bit through CA3, not C1, but CA3, the CA3 fields, dorsal ventral are very wide. Like, it's CA3 cells, innervates about a third of the hippocampus. So you can imagine there's a lot of interactions that we know that they want to CA3, that's integrating information from the emotion.
You talked about the time spiking, the difference between the cortex and then the hippocampus.
So we haven't typically recorded from the cortex in these class. We focus on the hippocampus. So this is something we keep doing, especially when they are instance so I cannot tell you what the cortical cells are doing. Something else.
And the position how the timing spiking, because that there's no, the same time spiking in the cortex and
Of course,
in the
yeah, right. I mean, you don't have plate cells per se, with some exceptions in the cortex. If you really want to see plates go, you're looking at the hippocampus. What you do find in cortical areas, like the cortex, are cells that are active during sleep that are related, in terms of spiking timing to some cells in the hippocampus. That you can find. There is no really place code that we can identify in the rat, right? Maybe different enormous in the rat that are bigger.
Yeah, but it's kind of interesting how it moves the signal in the campus in time, dynamical in time, not in time. How the signals between the cells, because you show only the spot.
Right.
But interesting is all by all the
Yeah, that's a good point, yeah.
dynamic factors. How move this part.
Yeah, so I found this pattern is very interesting. Seems to me it's like it's a statistical driven and you know, so another point that come to my mind is, you know, the right now very hot topic is automation, auto driving, like the vision based, like the Tesla, right? So they have to, like a robot, they have to navigate and find the right lane and based on the vision, based on the rules. Yeah, so I mean, this pattern, how can this, this whole set of experiment in your you presented can be, translated to current robotic consciousness and their navigation and their optimization. I mean, like it's like Tesla, they are rule based, but those animals, they, after a certain amount of experiments, they seem to know which route is best, like the traveling salesman problem mentioned many times. Yeah, so how can such pattern be, translated to?
That's a great question. I mean, I don't have a clear answer.
Yeah.
I collaborate with robotic people. I don't do it myself, right? But it seems to be that the most promising avenue for this, and also AI, I'll put one in a second, is the multi-scale, multi-field finding. So essentially, this says that, if you believe all of this, that, an efficient coding environment is at multi-scale, that you shouldn't think of the environment as being the car on the next lane. You should also think of the mountains or, I don't know, other things, right? So, there is a case to be made for the software to take into account space at multiple dimensions. All right, that's number one. The other thing that we thought about, and I'm digressing a little bit, is neural network, deep neural networks, when they do these pattern recognition essentially, and they can be full. You can generate adversarial attacks. I think it would be much harder to attack a network if you do it at multiple spatial scales and vertical scales, right? So, these networks have an input layer, right? And these input layers are convolving with a particular kernel dimension. They're all identical in some of the packages you find out there. What if now you have kernels, you have convolution kernels, and are on multiple scales like this, right? And it's random, or it's specific to a particular network. And it will be much harder to generate patterns of attack that would fold the network every. So there could be another application domain here is using convolutional kernels that mimic essentially the different scales of these events.
So you were gonna tell something, why it should be ambiguous?
Well, I mean, it's just intuitive, but when you, it seems to me that when you want to compute something, and you want to avoid being stuck in a local minimum, right? If you want to generate, you want to just, be able to generate some different results with the same input, and then, filter through essentially the one that are best. Maybe it's a good thing that cells, are not totally deterministic. From time to time, it just gives you something, different. Not an infinite possibility of outputs, but 2, 3, 4 times the number of cells, because you can be very creative. Maybe that's the key of creativity. It's how we create things. You go to sleep and then you have this multiple patterns, ambiguous things, and then you decide eventually, whether you use the context, right, to different kinds of computation to refine your output. In language, right, if I say pair, you don't know if I mean the fruit, or if I mean nighttime was two things, or if I mean something else, right? You disambiguate it. So I think we're actually wired in to deal with ambiguity.
Also, you can lose that cell and not lose that space in your.
Right.
Because you're only looking at a small number of cells too, there's lots and lots of cells. So there's
Oh, yeah,
ways,
right.
so like, once you get all of the input, you know exactly.
Oh, yeah, no, absolutely, right. So this is another way of dealing with this ambiguity, is to pull across multiple, but how do you do that is the question, right?
Right, like, but like the only overlap from these ambiguous maps is that one spot.
Right,
Yeah.
right. The difficulty here is the phenomenon of remapping. So now if you go to a different room, everything is changed.
That's-
Now think about the decoder who has been receiving inputs from overlapping cells in that environment.
Yeah, see, this is what I don't understand, like, you, say you have the exact same room, now put up different flags or whatever. Are the places are actually totally different now?
Yeah, right, they're gonna remap. And then what, you know, and the rat will still perform beautifully, if they compensate. It's a phenomenon that makes them recompute.
Yeah, I actually follow up to this. I was wondering about before already, like thinking about the Hippocampal stuff. People always talk about an environment, right? Like there is the environment, and there is a different environment. But like what, like if you have many small rooms connected by doors, are these different environments, or is it one large environment?
No.
Of cities? Like what's the definition? Right?
Yes, no, people have shown that, right? So if you teleport a rat from this room to that room, right, by hand, typically like this, they're totally confused. But if you let them go by themselves with a little corridor, they're totally fine. They know those are two different rooms, right? Right, so it's more than just the environment, it's the way you get to the way you.
You know where you are.
Yeah, you know where you are if you move, but the rat has to move to know, right? You can't just show the rat and say, this is the same one, this is the same room, right?
But in your multi-level example,
Yeah.
the levels look very similar.
They look very similar.
Like there are different possibilities, like the red could be, oh, I'm in this room, I'm remapping because it looks slightly different, and then I go up one level, oh, I'm in this room again, it has changed a little bit, I need to remap. Or it could be it's completely separate environments, or it could be one big environment that is sort of like, has wormholes where you can jump from one level to the next or whatever.
Well, I don't know.
Or do a counter instead, verbal.
Yeah,
Yeah,
I
okay.
don't know. I mean, if you recall place cells and you find that the place cell is selective to one level and never fires in the others, then it's hypothesis of, I know this is the same environment. If you see now that the same place cell is going to be recruited again at a different position on the second floor rather than the first, then you know, you're confused. But remember, there's cues around the room, right? And the rat is moving. It climbs, and it knows it's a different. I don't know, I mean, Sean is great.
Have you done population decoding? So like single place cell is not, it's ambiguous.
Right.
But if I put three of them together, do I get like more specific idea of where the that is?
That's a good point. I mean, we record from multiple cells at the same time.
Yeah.
Right? But we haven't decoded using multiple cells at this point. I'm sure we can do that.
I noticed, so there was a portion where you could get different responses from the same cells,
Right.
right? Have you looked at, basically whether or not that correspond, in the case of multi-field, neurons, whether or not those different like signaling patterns correspond to different place fields within that multiple field? Does that make
Sorry, I'm not sure I understand. So there is, as far as we can tell, there is no logic to where the subfields are.
Yeah,
This is what you're asking.
well, whether or not, so there's a firing pattern, right?
Right.
And it looks like it was delineated either in frequency space or just like time offset from the database, right?
Yep.
Do those delineations and firings correspond to one subfield versus another? No.
No,
Gotcha.
I wish, but no.
Yeah,
Meaning, if I give you a spike train of that cell, you cannot tell me which one of the subfield is coming
Yeah.
from. There's no way.
okay, cool.
I was just wondering if you noticed rats getting overwhelmed by just how many different environments you're having them learn over a specific time period, which would kind of allude to maybe a memory limitation.
Right.
Of like just how much spatial information we can hold.
Yeah, that's a good question. So first of all, I have to say that in all of these experiments, the rats are very familiar with the room. We spend a lot of pre-training time, right, making sure they can navigate in the middle of the room, they're not in front of the robot, rats and experimenters are pair bonded. So every student has 2 rats that we deal with for an entire year, so we use rats for a long time, right? Not that they need that amount of time to do the task, but we keep on connecting with our are they overwhelmed? So at the beginning, they are certainly afraid of the mega space, that's for sure, right? So we have a whole technique to ease them into navigating the living world. So, but after, I'm gonna say a month, maybe a couple months, they're totally fine. They go anywhere, they follow you, we sleep in the middle, they come to you. So I don't think fear is a big factor in the experiment. Right? Plus, remember, they're all positively motivated. They work for food. Right? There is no punishment of any kind. TSP, right? The training solution product. If they don't find the optimal path, we don't punish them. We just give them another try. Right? And they naturally find the shortest route. We don't train them to find the shortest route. They do it naturally. Right? So I don't think that's a big issue.
So there is no memory limitation in.
Oh, there must be some, right? There certainly must be some, but
The real world is a lot more complicated than.
oh,
these
yeah, for
things,
sure.
so They seem to do fine out there, yeah.
Yeah, and they, I mean, I can tell you that they remember things from day to day, from week to week, right? That, I'm sure, but I don't think we tap into the memory system so hard that, it's unrealistic or that, they're confused with that. I don't think so.
Have people found analogous megaspace things using VR?
All right, so there is definitely VR experiments,
Mhm.
right? Where the mouse is immobilized. Very interesting stuff. So my data is a classical example, UCLA. But you do find some differences. So theta is a little different. There are some different oscillations. We want it to be as natural as possible. Like have the rat actually move around, experience different, texture on the floor, etc., etc. So we want to slightly different. Now, do they find multiple fields? Yes, they do that too.
And what's something they can do that you could never do? Or do you think you can do everything they can do?
Oh, no, I mean teleportation, for example. Like all of a sudden, the environment changes.
But that's probably not very physical.
It's not, yeah, right. I mean, it's interesting. You want to challenge the system in many ways, right? You change the colors of things,
They have a lot more cells though, so.
Yeah, you can record for more cells. You can use different techniques. You can use, optical imaging, for example, when the head is fixed, right? And everything moves, which we cannot do, right? At this point. But I think we have enough cells. I mean, now we have, lots of probes that allow you to get, up to 1,000 neurons in conditions like that. A little bit improvement on the wireless system, and then we're set up to do all of this with recordings. Yeah.
Let's say you can have all the rats navigate Rome in the wild.
Right.
What would you expect? Is something you like to pursue?
Oh, definitely, yeah, the bigger the better. So of course, the first thing that we look at is the coding, these multi-scale, multi-field linear logic, At what point do we break down? So as Taylor said, if the Salk theory is correct, right, so bubble theory, right, at some point, you're gonna stretch a very small bubble, what's going to happen? You can't go infinitely small, right? The rats cannot perceive, the cell will not fire within, 5 millimeter. This is not possible. There is a limit. What is this limit? We don't know because, we don't have the space large enough. When the rat is confused, it has to do with PSP in an extremely big environment. What kind of other strategies are they going to use to find the rewards? Right? Now they definitely cannot use any boundaries, right? If you're in the rule, double assignments.
Yeah, there are no walls in the outdoors.
The landmarks, there are trees, bushes, big objects, right? But you're right, there's no walls. We need to have walls, otherwise we lose the rats with that. $50,000 implant in the head. The thing that we're really afraid of, so we looked at, fields, right, outside, is, eagles.
Mm.
Yeah, if they see a rat moving around, they just gonna dive on it, with the implant, and then why else?
Happy taught it to be in the open space afterwards.
Yeah, right now you see the rat in space, floating around, no, we don't want to.
You need to change the goal now.
What changes it?
Now it becomes 3D, yeah. right.
Changes it 3D, yeah.
Yeah, no, I mean, you know, we have to be reasonable, of course, there's gonna be delays, all the time, but I think we can learn a lot from larger environment,
But probably that the limitation is the sensor, right? Because you know that there's more than in the brain, because you know that all the signals you receive, right? The sensors you are, and then this is one of the main limitations in humans or in the animals, you know.
Sure, yeah, but I think we're not close to the boundary, we can go bigger and we can learn from larger spaces. We can do it. Yeah.
Well, one thing I expect to be different in the wild is, I mean, we really like that services,
Right.
floors and so on, but it's kind of artificial, right?
Sure.
You know, in the wild with all of the
Sure.
underbrush, whatever, like things that are not just obstacles on a like that door I can't move here, but it's like you need to kind of squeeze through
Yeah.
things that can move and so on, so.
Yeah, the question is, do you oversample with, place fields around these particular areas more than say in the middle of nowhere, right? Yeah, of course, yeah. All of these are great ideas, all we need is space.
Well, the desert.
If you're going to the desert, then make sure no eagles.
Well, leave the eagles, it's part of the natural ecosystem.
Oh, yeah, but the rats have extremely good visions upwards, right? They will see it and they will refuse to navigate. Right, so we actually tried this with a drone, believe it or not. A small drone, right, see if the rat.
An eagle drone?
No, it's just, the quiet thing, right? We can get the rats to get used to it, so even though it's very noisy, so rats don't like it. But they manage, right? I mean, the rats are amazing.
Thanks. Any questions online? Okay, let's thank
All right,
Jean-Marc
thank you
one
very
more